We're going to talk about the anatomy of the cell membrane, and believe it or not, understanding the cell membrane and how substances get across it is actually one of the most important parts of not only cytology, the study of cells, but of anatomy in general. In this presentation, we're going to focus on the cell membrane itself, the structure, in other words, the anatomy, and then in the next lecture, we'll actually start to talk about the exact mechanisms of how substances cross over. So you may recall that the cell membrane is a phospholipid bilayer. And in the word phospholipid is lipid as in fat or oil. If you think about film of oil or soap bubbles and all the swirly purple and green colors, that's what you get in a lipid or an oil. So a plasma membrane is made up a lot of the same stuff as soap bubbles. So that brings us to a mystery to solve about bubbles. If plasma membranes are so much like the substance that they're made of is so much like a soap bubble, can you start to think about some of the ways that they behave differently than soap bubbles, and how are we going to figure out how they're structured in order to do that? So here's some mysteries to solve. How do we get substances through the soap bubble? I mean, why don't they just pop? Here's another one to think about. Do you remember how many different types of cells there are? There's over 200 different types of cells. Now, can you picture the shapes of cells? Imagine any specific cells that you can think of, any specific cell types. We looked at some red blood cells and some nerve cells. Maybe you can picture different types of muscle cells. Now, imagine going on a walk or a hike. It could be in the park or the woods. What kind of stuff do you run across on a walk in the woods or the park that has the similar kinds of shapes of all the different kinds of cells? Trees, pine cones, sticks, blades of grass, even the fluffy tufts of dandelion flowers. Have you ever seen bubbles shaped like trees or pine cones or dandelions? And if not, how could you build cell walls that are made up of a lot of the same lipid stuff that soap bubbles are made up of and still get them to be in all those different shapes? So there's some things to think about as we learn about cell membranes and how they are like soap bubbles and how they aren't like soap bubbles. So we're going to start off talking about what does a plasma membrane, or in other words a cell membrane, what does it do? What is it designed to do? What are its jobs? You probably recall the term selective permeability. That means that cell membranes are able to let some substances through and keep other substances from crossing. They are choosy. They're able to decide what substances cross over and what substances cannot cross over. This actually reminds me of, and people will sometimes use the analogy of walls on a building. So here we're looking at a cabin, which actually reminds me of a motorcycle road trip I took in BC, and it was absolutely stunning country, just beautiful back roads, all kinds of evergreen trees, fir and Douglas, and really tall old growth, just amazing. And there was this little what appeared to be a deserted log cabin on a side of a little back road in a field, kind of similar to this picture, although this is not a picture I took of it. And I saw some kind of rustling in the grass, and so I pulled over, and these incredibly cute, totally adorable, 2 little black bear cubs are scampering through the wildflowers, and they're leaping through the flowers, and they look almost comical, just completely adorable, roly-poly and jumpy and springy. And they went over to the cabin, and they were exploring and sniffing around, and then one cub put its paws on the window, the open, broken old window of the cabin, and then put its paws in, and stuck its snout over, and then hopped right in. And then the other one followed suit, and then they were playing around like this, hopping in and out of the windows and going in and out of the doors. And I was just on my motorcycle by myself, so don't worry, I was keeping a good eye out in case Mama Bear was around, because they get pretty fierce if anybody gets near their cubs. But anyway, the point of this story, why it reminds me of cell membranes being selectively permeable, is you have to be careful what you're going to let inside and outside of the cell. And in the case of a cell membrane, it separates the intracellular, in other words, what's inside the cell, and extracellular, what is outside of the cell. And which chemicals are allowed in and out are very important. And hopefully, from the previous slideshows, it's starting to sink in why membrane selectivity is critical to the fluid medium inside the cells. First and foremost, there's a balancing act that happens in the body of all kinds of chemical levels and temperature, and maintaining certain ranges of chemicals and temperature in a certain fine-tuned range, despite changes inside and outside the environment, is one of the most important concepts in physiology. And hopefully you will know this term inside and out, and its many applications in physiology, and that is homeostasis. Homeo what? Basically just keeping the chemical levels, the temperature levels, the pH in a certain range despite the changing environment. You may remember that there's not only so many chemicals sloshing about, that it's just like the levels of chemicals in a pool. But one thing that's different from a pool is a cell being a living organism. It actually is carrying on lots and lots and lots of different chemical activities, and all those activities are called metabolic activities. And all of those create waste products. So of course you're having to handle those waste products. So just like you have the changes coming from the outside, you also have the changes coming on from the inside, like the metabolic activities creating waste. And to go back to the analogy of the pool, you wouldn't want to swim in a pool of pee, just like the cells wouldn't want to be full of metabolic waste like uric acid or urea. This reminds me of swimming in public pools. Have you ever gone swimming during the times where there's lots of babies and toddlers? It's kind of like Russian roulette. One day, or one minute, it's like all sunny and sunshine and happiness, and then the next minute you hear the scary shark music. And then the lifeguard yells, "Evacuate!" and they have to clear it out and go fishing for poop. Everybody out of the pool, and then they're fishing for poop and dumping in chemicals, lots of chlorine. Nobody can go in the pool for a while. So that is homeostasis in a public pool. Scary. But okay, so this what gets inside and outside of the cell is huge. It's critical to the cell's not only ability to live, also to be healthy, but also for it to carry on its many, 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 many functions. So with over 200 different kinds of cells, they're not just like these little round soap bubbles floating around, just generic cells, but they all have specific jobs to do. Think about any specific jobs you can think that cells have to do, and a lot of those specific jobs are going to actually involve the cell membrane. So the cell membrane is cool and critical not just to decide what gets in and out of it, but actually because it has to do a lot with specific functioning of the cells too. For example, communication. Now this works with a lot of the communication systems in the body. For example, the nervous system, using nerve cells to conduct electrochemical signals. Both the electrical part of the signal conduction, which is, like kind of like a flash of lightning, or it's like knocking down a long pile of dominoes, and the cascade reaction and the quick sending of the signal, the electric part carries out on the cell membrane, through the cell membrane, and because of the cell membrane. And it is an active part and reason of why that electrical signal happens. So it's not just like a passive, envelope. And then at the end of the nerve cell, it has to spray out little vesicles, secrete little vesicles that have chemicals in it to send signals. And that ability to send those chemicals out of the cell, that also is very much relying on how the cell membrane does its job. There's communication between, so not just the nerve cells themselves, but the communication of all different kinds of cells relies on the plasma membrane. For example, when hormones, which are chemicals that send signals, when those hormones go floating around in the body, cells have to be able to understand or translate what those messages mean. And so in order to receive those messages and interpret the messages, that is also relying on the cell membrane. So there's special little proteins in the cell membrane that have to do with receiving those signals, those chemical signals. And there's other ways too, and we'll see this as we study each system of the body. And another function that the membrane, cell membrane serves is also as sort of identity markers, so that the organism and its every cell is has a way of identifying itself as the body's own cells. So, for example, if you had foreign substances like bacteria, viruses, fungi enter the organism, they, in most cases, are not going to have the same little identity markers. Now, some of them come up with, tricky mechanisms of trying to imitate those identity markers so that your body doesn't recognize them as invading substances. But what your body tries to do is have these little proteins on the surface of your cell membrane that are like little flags. They're like countries with their own flags saying, okay, this is Sweden, and each one has its own flag. And many people know of the blood typing system that uses A, B, and O. That has to do with those markers. So it's important to understand that the cell membrane is a semi-permeable phospholipid bilayer, and we're going to take a look at the actual structure of this, the substances that actually create this, and their chemical properties, because the next thing we're going to learn is how substances actually cross this membrane, and these the way that substances cross is very much dependent on the anatomical structure. So we're going to kind of break it down and look at the different elements here. So first of all, starting off basic, semi-permeable means that some things can get through. And phospholipid refers to these molecules that have fats in them. They have a phosphate side and a lipid side. And this is going to, and these arrange themselves in a bilayer, which bi means two. So it's a double layer, like two sheets put together. And these parallel sheets of phospholipids actually are self-arranging, or they arrange themselves. And we'll take a look at the fluid mosaic model next and see how that how they can actually move around. But looking at this diagram here, the little circles are the phosphate part of the lipid, or the phosphate segment, and then like a ball, if it was, and then there's like two little like legs or antennas on the ball, and that's the lipid segment. And having these two different chemical sides, it ends up that there's a charged or polar side, and that would be the phosphate side, which is then water-loving because it's charged, and then the water-hating or hydrophobic side that is non-charged, the lipid side. Let's and these phospholipids, this is the part of the membrane that is like the soap bubble. But you can see in the diagram, and in this particular diagram, it's in pink and purple, there are all these things inside the phospholipids, and these are mostly proteins. So there's a bunch of proteins embedded within the lipids, and we'll take a look at what these different proteins do and how they're structured in just a minute. But let's actually talk about this in terms of the fluid mosaic model. So the cartoon-like diagram we just looked at might give a rather static impression, or if people use analogies like walls of a building or something, you might think of the cell membrane being sort of rigid. But cell membranes in animals are not made up of walls like in plants. In plants, the cells actually do have walls, and it's basically because plants don't have their own little skeletons, and so each little cell having more of a firm wall structure actually provides more of the skeleton of the plant. But the fluid mosaic model of the cell membrane is basically looking at the different parts of the membrane and how it actually moves around a lot. So the phospholipid part, that's like a soap bubble and it has a consistency of oil, those little phospholipids are self-orienting because there's a positive charge, there's a charged side and a non-charged side. If any of them get poked around or disrupted or moved, they can kind of jostle around or even float around, and they'll reorganize themselves. So if a couple of them get knocked out of place or nicked out of place, because of the part that's charged and not charged, they'll reorient themselves. But within that, they can actually move around, and if they were cut, they would actually reseal themselves. And within the soap bubbly kind of phospholipid part are little rafts that are like lily pads. And these lily pads add more structure and stability to the membrane, and also provide areas so that materials can travel through the membrane and make it more stable and make channels to go through. So some of the materials that are in the little lily pad rafts or lipid rafts, there's a lot of, well, the first thing you see on the list says phospholipids, and that might actually be confusing because didn't we just talk about the phospholipids making up the fluid part? Well, yes, it's true, and this might be more chemistry than you want, as much of a lot of what I said might be. So for this next little teeny bit, you can just go la 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 la, I'm not listening to you. But so the little balls with the little legs on them, the little phosphate heads, and then the little lipid tails. If you want to make them more fluid and movable versus more close together, there's a way you can do that chemically, and basically it's to make it saturated or not saturated. It has to do with how many of the little molecules are, remember when we talked about that rule of 8, and if those electron pairs are all, if the electron shells are all full, how they're more stable and more happy, and if they're not full, then they get a little irritable, they get unstable, they're looking desperately for something to connect with. Well, a lot of what the body is made up of is carbon, and so when you talk about things like fats being saturated or unsaturated, has to do with how many of the little connections that could be made with carbon are made with carbon, and if there's any of them not made. And anyway, you don't need to remember which is which, but basically saturated versus unsaturated is whether or not you have all the little carbon connections made, and that can determine whether these little phospholipids basically have a kink in their tail. So whether they're, you know, like the little person with their legs standing out, or a person with their legs standing straight. And that would determine, I think that one's more like a space alien, but anyway, that'll determine whether they're spaced further apart or held closer together. And I am certainly not going to make you memorize that, but again, sometimes I give you more detail than I will test you on, because while you may sometimes just want the simplest version of information to memorize possible, sometimes it's actually useful to understand the chemical composition in a little bit greater depth, because then you can actually form a picture in your head of what's going on. And then some of the other things in the little lily pad rafts are cholesterol and sphingolipids. It's not very often you get to say sphingolipid, so I encourage you to take this opportunity. You're among friends. The cholesterol. People often think of cholesterol as just a bad thing. Cholesterol is actually necessary in your cell membranes and other places in your body, just like there's good fats. But one thing the sphingolipid cholesterol part of the lily pads do is they basically they're like quilt-like patches in a quilt, and they happen to hold stuff together more, so they have a more stable structure, like lily pads floating in water. They would be like the lily pads. But aside from just holding stuff together, they can do something else really cool, and they can actually hold the proteins in place. So inside a lily pad, we could have one of these gorgeous flowers and pretend that the flower, I can't draw flowers. Definitely can't draw flowers with my little marker here, but pretending that one of these beautiful flowers was inside the lily pad and that was a protein, then they can do cool stuff like be little channels or gateways. And there are actually stems going down here from the bottom of the lily pad, just like when Thumbelina gets deserted on the lily pad. I think it's by a dragonfly, and then the fish are so sad for her that they go and they start nibbling. I can't draw a fish either. You'll just have to imagine fish. And the fish are all sad for Thumbelina, so then they nibble. And this right here in the body, since I'm just telling a story of Thumbelina here, This stem of the lily pad would be going through the membrane of the cell, and this would be a channel where proteins could get through. But in the story of Thumbelina, she gets deserted on this lily pad, you may recall, by a dragonfly, I think. And I guess it depends who tells the tale. And the fish are sad for her because Thumbelina is so awesome. And so they nibble at the stem and free her, and then she gets to take this awesome water rafting trip, white water rafting trip down the river on lily pads. It's pretty cool. Let's take a look at a nice animation of the fluid mosaic model, because a lot of times when you look at these diagrams in textbooks and all, just limited by the two-dimensional nature of paper, there's only so much of a picture that you can get of what this actually looks like or how it actually works in the body. And this little animation I think is pretty cool, just to give you a little bit of a feeling of what the phospholipid bilayer is really like. So here, if something bumps into it, it is actually very flexible, and even when these little phospholipids get knocked out of place because they're positive, or they're charged and non-charged sides, they'll scoot right back in line. Oh no, I'm getting seasick! But it's like that. They move around. And what this animation does not show is that they not only go up and down like waves, but they can actually float around and move around left and right and squish about. Now we're seeing some protein channels, some lily pads letting stuff float through. And we'll talk in the next segment, the next lecture, about how this happens. But those little protein channels, those green little lily pads, can move about. Here comes one floating now in a sea of phospholipids. What does limit it from actually just floating about all the time are the cholesterols, which anchor, as well as there are also proteins that anchor. So there are some things anchoring it in there. All right, so let's watch another super quick video that's going to tell you some real basics about the cell membrane.
Hi, my name is Mary Poffenroth, and I'm an adjunct professor of biology. And today we're going to be talking about the plasma membrane. The plasma membrane separates the internal environment from the external environment. It kind of acts like border control, controlling what comes in and what goes out of the cell. The plasma membrane is made up of mostly proteins and special lipids called phospholipids. These phospholipids arrange in a double layer or bilayer arrangement. The top and the bottom of this bilayer arrangement are water-loving or hydrophilic, and the inside of the tails is going to be water-hating or hydrophobic. Think of it kind of like an Oreo cookie, where the brown chocolatey cookies are going to be hydrophilic or water-loving, and that creamy center, that's going to be water-hating. So let's think about it for a second. Why would a cell want the edges that touch the outside as well as the inside of the cell to be water-loving? Let's use humans as an example. Humans, like most creatures on this planet, are mostly made of water. Now, you wouldn't want a cell floating around in a bunch of water that was water-hating, right? Most plasma membranes also have special proteins embedded in their phospholipid bilayer. These special proteins help with the transport of molecules across that membrane, and they serve other special functions, like giving the cell shape, as well as adhering to other cells to create tissues. Thanks for watching, and if you want to learn more about this subject, click on the link below.
All right, and while we're at it, we're going to watch one more quick video. And I'll show you a master link here because this is just about a 3.5-minute lecture of Carl Schuster, who I think his video lectures are excellent. Some of them are a little bit more detailed than I test you on, but again, I think a little bit more detail sometimes actually explains things better. So I like his videos so much that I put a link to them on the personal settings of my Canvas page. So rather than put links to all his individual videos in each and every one of our units, I just put a link on my personal settings, but I highly recommend that you check them out, and this is just a sample of one of them. The only thing I don't like about his videos is that the screen recording system he used is currently not iPhone friendly, so because of the flash player it uses. So you currently cannot watch them on your iPhone unless I'm recording it through my player, and then you can see it. But if you're using his links, you need to use a computer, and I would imagine his college will probably try to change that if they can, but it has to do with how they recorded it. So let's take a look at the proteins and cholesterol inside the cell membrane. There we go.
Another molecule we commonly see embedded in the plasma membrane is cholesterol. Cholesterol, you will recall, is a lipid molecule. In fact, it's a steroid. And it is therefore soluble in other lipids. Well, I want to describe the role of cholesterol to you as that of rebar. Rebar are these metal poles that we put inside of concrete in order to help concrete stay together. The concrete molecules don't stick to each other very well. Well, they do, but they tend to fall apart on the outside edges. By putting a structure inside of the concrete, this allows the concrete molecules to stick to something inside. Therefore, they tend to stay together better. And this is the role of cholesterol. It floats inside of this jello-like matrix and stabilizes the membrane by allowing these neighboring molecules to stick to it. Besides carbohydrate and lipid molecules, we also see a lot of proteins embedded inside the plasma membrane. As a matter of fact, proteins make up more than half of the membrane by weight, and we can categorize them in two types. Some of these proteins are what we call integral proteins. In other words, they are inserted or built in directly into the plasma membrane, as you're seeing here. You know what it means to be an integral part of a team. It means you're a basic part of the team. Some of these proteins face to the outside. Some of them are inserted deep inside the bilayer. These integral proteins can act in several ways in the cell's physiology. Sometimes they are receptors and they act to attach to other molecules. Sometimes they're going to be used as recognition markers for the immune system. Sometimes they can be anchors, whether on the inside of the cell here, anchoring the cellular skeleton, or on the outside, anchoring one cell to another. They can also often act as carriers or channels. We're going to talk a lot about integral proteins as the semester goes on. Your author gives you a table outlining some of these basic functions: receptors and enzymes and channels and identity marker proteins. Here we're seeing an anchor for a neighboring cell. These are called cell adhesion molecules, or CAMs, a molecule that anchors to a molecule in the membrane of a neighboring cell. Another classification of proteins are those that are not embedded in the plasma membrane, but rather sit on the outside. Many of these on the extracellular side are enzymes. Many on the intracellular side are anchors for the cytoskeleton. So here you're seeing a peripheral protein attaching to the cytoskeleton inside of the cell.
I'm going to pause him right there because I don't remember if he's going to mention this or not, but this is a pretty cool diagram here on the bottom with all these little tubes like the Doozers build in Fraggle Rock, or like scaffolding. Because I am just going to guess, if you ever think about the inside of a cell with the organelles floating around, if you ever give it that much thought, I would imagine you might think about these little organelles just floating about in a sea. But there are actually little tubes of scaffolding. There are little filaments of protein that help hold them in place. And this is important to hold elements close to each other because there's incredibly complex cascade reactions of chemicals going on, and having things in the right place and then having these filaments kind of guide the process is very important. If everything is sort of very time critical in order to get these chemical reactions to occur, you even need to have these things close together so you can kind of position things at. And these little filament proteins, they also connect to the cell membrane itself.
This occurs, of course, within the intracellular matrix. However, these proteins can attach to fibers in the extracellular matrix as well. This helps to give the cell some shape and to stabilize it in place. Another function of the cytoskeleton you're seeing here is that it does hold the internal organelles in place. They are not just floating around freely inside the cytoplasm.
All right, so he just said a lot of stuff that proteins do in the cell. Let's take a look at it a little teeny bit slower. So the proteins that are inside the cell membrane can basically be divided into two different kinds, integral and peripheral. And basically that's just saying which proteins go right inside and in many cases all the way through the membrane, and that would be integral. They go inside the membrane and oftentimes all the way through, versus on the surface of the membrane and like sticking out of the membrane. And those are called peripheral, like on the periphery or the outer edges. So these proteins, whether they're going through the membrane or on the outside of the membrane, they make up about half of the plasma membrane, and that is by mass, in other words, by weight. Make up about half the plasma membrane. So about approximately roughly half is the phospholipid soap bubble, and then about half of it, by weight anyway, although these proteins are going to be a lot heavier than these phospholipids, they're going to make up about half of it. So let's take a look at some of the jobs these proteins have. They have a lot of big important jobs. A lot of the proteins that go through the cell membrane are involved in transport. So a lot of times they are channels or pores, which are, if we go back to the analogy of the log cabin with the windows and the doors wide open, they would be like windows or doors. So they're basically pores to let bigger stuff through, like bears. But and some of the ways the body can determine and the membranes can determine what gets through or not are just how big those pores are. And we're going to look at other ways they determine what goes through or not. But it'd be like putting a, well, this wouldn't work for bears, but let's say you put a screen door on your house door, and now you have this nice little fine mesh. That fine mesh is going to keep, most everything outside, like the mosquitoes. And but let's say your dog accidentally scratches a hole in your screen door, and now you have a bigger hole in that screen door. Well, now your mosquitoes and other stuff can get in your screen door. Well, so some of how these protein channels work are as simple as just what size hole is in the screen mesh. But then, of course, as you can probably guess, there are other ways you can determine what goes in and out, because that really wouldn't be that selective to just have different size holes. And if it was big enough, if the hole was big enough, stuff would get through. You have to have other ways to choose what gets through or not. Now, some of you may know a thing or two about chemistry and physiology already, but whether you know something or not, go ahead and think about this like a puzzle you're trying to solve. if you wanted to think in chemical terms and keep some stuff out and let other stuff in, what did we look at when we looked at the chemistry of water that was one of the main determinants of how stuff worked chemically? Well, a lot of stuff works chemically based on charges and whether it has any charge at all or whether it has a positive or negative charge. So a lot of times these just can be like voltage gated or ion gated channels to whether they're going to let positive or negative stuff through. And this is totally like, you know, the game. Red Rover, let phosphates come over. Yeah, no, we didn't play that version as kids. It was a little bit different. Where was I? Oh yes, channels, pores. How else do the integral proteins work? They can also bind with substances, so certain chemicals can connect to the proteins, and we'll take a look at this in detail next lecture. They can also be receptors for different chemical messengers like hormones. One thing to start really getting a good grasp on in your mind and forming a mental picture is that proteins are really, really gigantic molecules. They have lots and lots. They're like this big giant string. And with this big giant string, you're going to have a lot of areas that are all kinked up and twirled on each other like a big ball of yarn that got all snarly. Your cats have been playing with it and it's all a big mess. You're never going to separate it. Or sometimes they'll end up forming like a little twister tornado. And you get all these crazy three-dimensional shapes when you're talking about proteins. And the shapes and where there's little positives and negatives and where they're looking for carbons, and that's going to determine a lot of what interacts with them. The proteins that are peripheral, on the periphery, sticking on the outside, they have some different jobs. They can be the little filaments, so the little basically like squiggly lines that can hold one cell onto another cell or hold together different parts of a cell, giving it some stability and some structure. They can be enzymes, which is basically, and enzymes are an important concept. Enzymes are basically a chemical that helps other chemical or catalyzes other chemical reactions to happen faster usually without being changed in structure themselves. So, I mean, we have just way more chemical processes than anybody could count or comprehend going on at any one time. And a lot of these things need to happen really quickly, like when you're sending a nerve signal or firing a muscle. And so a lot of times you need to catalyze or speed up chemical reactions. I think this last one is pretty cool. Your cells are actually sugar coated, so you have sweet little cells. And what it actually is it's a combination basically of a protein and a carbohydrate. The glycocalyx is this little purple antenna on the little pink mountains. That is the glycogen or the glyco, the sugary part, is the little purple antenna sticking out, and then it's stuck to a protein. So it's like a sugar on top of a protein. But these are those little markers or those little flags, the little identity markers of your body, identifying its own cells, which are a very important part of the immune system. All right, so that's the basic structure of a cell membrane. And let's see. I gotta get out of drawing mode here. Excuse me, we can't move on. I'm stuck till I get out of the drawing mode. There we go. Okay, so now that you know more about how a plasma membrane is formed, the phospholipid bubble-like part making up about 50% of it by weight, and then proteins and cholesterol forming the little lily pads and filaments, channels. Now that you know more about the cell membrane, if you think back to some of our questions about bubbles and how we could take something like a soap bubble but give it more defined structure and shape to have the shapes of our over 200 different kinds of cells, and then how could we actually get substances to travel through a cell membrane. So let's take a little look at a little science experiment. And then after we take a look at the bubble experiment, just what's next? The next slideshow, we're going to look at how substances actually cross over the membrane. But let's take a quick look at a little lab. This she's mostly using a bubble soap mix with some glycerin, by the way.
Or you can say, like, here you go. Find 3 ways to get that pencil through. Way number 1. Take the plastic tubing, submerge it in bubble solution, so that when we pull our contraption up here, we can actually put this through, and the pencil slides on through.
All right, and so in that little part of the experiment, what is the little tube in the cell membrane? What acts like the little tube, and why did she have to put the soap solution on it before she put it in there?
Without breaking the membrane. So the membrane's still intact. That's the first way of getting the pencil through without breaking your membrane. Alright, second way, using the string. The easy way is just to kind of like place the string right on the top so that as you pull this up, now you see the string is embedded in there. You can actually poke the middle out of the string. So you see that we have this hole here where the pencil can pass on through. Oops. Oh, yeah.
And again, think about in cell membranes, what is acting like the string embedded in there? What's the material made out of, and what's the name of it?
You get the idea. And the cool thing about showing this or having the students do this is you can show that, you know, even though you now have this hole, this opening in your cell membrane that allows you to pull things through, you can take your paper clip, you can actually pull that string out, and you see that the membrane closes right behind it, kind of demonstrating endo and exocytosis of the.
So that she mentioned endo and exocytosis, which we're going to get to next time, but it also demonstrates how when the membrane itself is disturbed, that with the fluid Mosaic model, the little phospholipids are self-orienting, and they will go back into position, and they will reseal themselves when torn.
Cell membrane. Alright, and lastly, and the most fun for the students, they actually take the pencil, you just submerge it in bubble solution, and they find out that anything that is made of the same properties will actually pass through the membrane there. Now,
and think about why is that? Why, if you coated the material in the same material, why could it pass through? And in the body, how would cell membranes be structured for that to work and for substances to cross through? And when you're thinking about that, think about the hydrophilic and hydrophobic and positive and charged sides and non-charged sides and lipids and all that.
It's usually at this point when they figure this out that kids get the crazy zany idea that, you know what, if I can get that pencil to do that, I bet I can get my hand to do it. And the answer is, yeah, they totally can. So you can actually take your entire hand and start placing it through and pull it back without popping the membrane. I've had kids actually get their whole arms through. It's a complete mess, But the great thing is that it's soap, which means that after the lab, your kids have completely washed all your tables, which is great. Things that are made of the same material, such as the lipid, will actually pass through the cell membrane without any need of a protein channel, okay, or endo or exocytosis, such as with using the string. So like I said, this is a great way to help kids understand the properties of the cell membrane. It's a fun lab form. They really, really enjoy it, and they really have a good.
All right, cool. Thank you for your time, and hope you spend some time thinking about how the cell membrane is structured, the phospholipid part, the protein part, the cholesterol part, and start to think about how we're going to get substances across and why that is so important. And then start thinking about the other functions of the cell membrane too, because as we study each system in the body, you're going to see that the cell membrane is critical for functions throughout the entire body. So sometimes these basic concepts at first, anatomy and physiology kind of builds in layers, and when you first look at a concept, it may not quite make sense, but these are kind of the important critical building blocks, the foundation that you kind of have to spend time grappling with them with your mind. And until you're on a walk and random things like trees start to remind you of anatomy and physiology, you're not thinking about it enough. So just start to think about it as you're going about your day. And there's so many things when you're making your dinner or making a cup of coffee. I mean, there's chemistry, there's biochemistry, there's anatomy and physiology in action. You could start to think about how all these systems in the body actually work. And then anatomy and physiology comes alive, and it's completely amazing. Thank you for your time.